Hi, I'm Tim Avis. I'm an investigator at Summit Therapeutics, and I'll be sharing our data on the mechanism of action for ridinilazole. C. difficile infection is a global problem and brings a significant economic burden to healthcare systems. Until recently, the precise mechanism of action of ridinilazole, an investigational antibiotic in phase 3 trials, had yet to be elucidated. We have evaluated DNA binding as a potential mechanism. This line of investigation was explored due to the structural features that ridinilazole shares with other bisbenzimidazoles, such as Herx 33258, which has previously been shown to bind to the minor groove of DNA. Both molecules share an overall crescent shape which aligns to the conformation of the minor groove of DNA. We can now present our data that shows ridinilazole binds with high affinity and sequence specificity to the minor groove of DNA. The interaction occurs at sub-MIC concentrations and is predicted to have consequences on cellular functions within C. difficile. We first used ultra-pure DNA. Here we titrated the DNA with a fixed concentration of ridinilazole, a concentration where the intrinsic fluorescence of ridinilazole was at a basal level. We see a clear dose-dependent increase in fluorescence upon association with DNA. Next, we wanted to show ridinilazole bound to a more relevant DNA substrate, C. difficile-derived DNA. We anticipated that we should show this through UV visualization following gel electrophoresis. Here we incubated the DNA with a titration of ridinilazole above and below the MIC. Fluorescence from ridinilazole complex to DNA can be seen as low as 1.22 nanomolar, which is over 100 times lower than the MIC. Densitometry allowed us to semi-quantitate the fluorescent bands, which you can see in figure 2b. An apparent dissociation constant of 87 nanomolar was determined from the plot, again significantly lower than the MIC. To test sequence specificity, we used 3 different DNA polymers which have been widely used to study drug interactions and DNA structure. These DNA polymers consisted of either AT-rich DNA or GC-rich DNA. We can see that when the DNA was titrated with ridinilazole, only DNA containing adenine and thymine bases shows an increase in fluorescence. No increase in fluorescence can be seen with DNA containing guanine and cytosine. This data shows a propensity for ridinilazole to bind to AT-rich DNA. To study the binding affinity of ridinilazole, we chose 2 different short double-stranded DNA oligonucleotides that were predicted to have single-site binding. Based on previously characterized data for the minor groove binder, Herx 33258. We used a titration of the oligos with several fixed concentrations of ridinilazole and determined the KD for each concentration. From these dissociation constants, we used a linear fit to extrapolate the true dissociation constant. Potent binding at ridinilazole concentrations significantly lower than the MIC were observed, with a dissociation constant of approximately 20 nanomolar determined for each oligo. Finally, we chose one of the short double-stranded DNA oligos to take forwards for X-ray crystallography studies with ridinilazole. The solved co-crystal structure of ridinilazole and the short double-stranded oligo can be seen in figure 5. Structure was determined to a resolution of 2.2 angstroms. Excellent compound density was observed for ridinilazole and can clearly be observed occupying the minor groove, with proximal nucleotides to ridinilazole being AATT. We have shown that ridinilazole demonstrates potent binding with AT sequence specificity to the minor groove of DNA at sub-MIC concentrations for C. difficile. We believe DNA binding is the primary mechanism through which ridinilazole exerts its bactericidal activity in C. difficile. We are currently carrying out further studies to determine the downstream effects of ridinilazole binding DNA within C. difficile and which cellular functions are subsequently affected.